Well, hello and welcome to Wellness Wednesday. My name is Sally Duplantier, and I am the host of Wellness Wednesday, and I'm also the founder of a company called Zing. My mission is to help people live their best life longer through better nutrition, more movement, better sleep, and more mindfulness. And I am delighted to execute on my mission through these free programs and to welcome Dr. Christopher Gardner here today.
Happy to be here.
These programs remain free to the community because of generous sponsors. And I'd really love to thank Jacqueline DuPont. She's a recognized thought leader in the field of gerontology. She's a philanthropist. She's the co-founder and CEO of Assured in Home Care. And at the start of the year, she generously wrote a check to cover 6 months of expenses to keep our program going. So much appreciated. Now, I am delighted to welcome back Professor Christopher Gardner, and we could spend the whole 45 minutes just talking about his background, but I'll try to be brief. He is the Renborg Farquhar Professor of Medicine at Stanford, and he also leads the nutrition studies at the Stanford Center for Prevention Research. Because of the rigor and quality of the design of his human intervention trials, he's recognized as a leader in nutrition science research. And he currently serves as the vice chair of the Nutritional Committee of the National Heart Association. One more thing that I will add is that he was also my mentor when I did an internship at Stanford last year. And so it's just a delight to welcome you back, Professor Gardner. Thank you so much.
And I just wanted to point out, if anybody couldn't tell, Sally picked a really old picture of me that looks very young. But Sally, I noticed the shirt was almost the same color. And I wanted to let everybody know I have bought a new shirt since that last picture.
I'm glad we cleared that up.
I think people were worried. Okay. So here we go. I am going to share my slides now, correct? Okay.
Yes.
Share screen. It is, I'm honored to be here for the second time talking to all of you. Okay, whoops, I looked through my slides and now they're at the end, so now I have to get them at the beginning. Okay, we'll get back to the beginning. We're going to jump right into this. We're going to save time for questions. And if you want to write questions in the chat, when I'm done, I'll be able to review the questions in the chat. And Sally, I'll bet you'll be able to look at some of them. I can't see the chat as I'm presenting, but you may be able to look through them and find some questions to queue up for me at the end.
I will definitely queue up questions for you.
All right. Well, I have the honor of being at Stanford. I've been here for about 30 years. And most recently, my career shifted by becoming friends with Justin and Erica Sonnenburg on my campus, who are microbiome experts. And I have called myself a feeder and a bleeder for about 2 decades. I get a lot of people to eat things, and then I take blood samples and look at their insulin and their glucose and their cholesterol. But now I'm a feeder and a bleeder and a pooper. And so now in all my human studies, I get poop. No shit, I have all this stuff in the freezers where they've donated fecal samples and they analyze them for me. They actually are terrified of working with humans. They usually work with mice. And I think poop is icky. So it's actually like 2 pieces of the amulet come together because I do what they want to do and they do what I don't want to do. And so a lot of what I'm going to share with you comes from my work with them. I'm going to guess the topic of the microbiome is familiar to a lot of you. I see it in covers of magazines all the time, this just being a quick selection of some of the magazine covers that I've collected, and plenty of books. I think my arrow shows up when I talk, right, Sally? So I'll just hover over the good gut, which if you really want, one of my favorite books on this topic was written by Justin and Erica Sonnenburg. This is a great, very practical overview. They even have menus at the end and recipes. So here's an outline of what I'm going to present for you today. And the first thing I thought I would do is just clarify 2 terms that I find the public tends to think might be interchangeable: prebiotics and probiotics. So a real quick overview on what those 2 terms mean in this field. So here they are. Here are the prebiotics, which basically just means fiber. So prebiotic, the biotics part refers to the bacteria in our bodies, and that's this hot topic, bacteria all over the place. But the highest concentration of the bacteria is in our colon. And the very definition of fiber from a nutritional perspective is we don't have the enzymes to break it down and absorb these carbohydrate-rich sources. And so since we don't break them down and digest and absorb them, they go to our colon, where the bacteria, the trillions of bacteria, hundreds of different species, have a heyday eating them up. Unless you took a pill, because there's plenty of pills that have fiber in them as well. And so here's just a small selection of pills, none of which I'm promoting. I'm just simply trying to show you how many pills are out there. Now, this would be very different than probiotics. So probiotics simply means live bacteria in the foods you're eating and the beverages you're consuming. And we did a study on this, and we try to give people a lot of fermented food. These are about the only ones Americans, as far as I can tell, are willing to consume. There are some others. There's natto, that's Asian, that I find a lot of people are not willing to eat. If anybody wants to put in the chat if they've ever had natto before, which is pretty slimy, smelly, fermented soybean. Yep, we did a study and we were able to get people to drink kombucha, drink kefir, eat yogurt, sauerkraut, and kimchi, but not a lot of other things. So all of these have live bacteria in them, unless they fooled you and they made sauerkraut by just taking some cabbage and throwing vinegar on it. It would give you the same taste, but it wouldn't actually have live bacteria in it. So one of the things that you can look for is a term called CFUs. These are colony forming units is what it's called. And it's kind of silly. Look at this. This pill has 40 billion CFU in this tiny little tablet. So there are billions and billions of them and trillions in your body. So I don't actually know what the right number is to try to get. I'm just trying to help you differentiate prebiotics and probiotics to start with. And there are plenty of these pills out there. Okay, so now we've got that terminology to start with. Now let's talk about nutrition meeting microbiology. Now these slides are pretty much from Justin Sonnenburg. I tweaked them just a little bit. Let's start out with your intestinal tract, your small intestine and your large intestine, and there's circulation floating around on both ends. And let's say that you are going to eat something really simple, like a piece of cake that really just has sugar and starch in it. Well, our human genome has about 20 different genes that create enzymes that will degrade these so they're small enough to absorb and put in the circulation. In comparison, the microbiome in our colon has more than 10,000 genes to make a wider range of enzymes to break these things down. If all you ate was cake, not much would end up going to the microbiota because it would all be absorbed here. And look, don't they look sad? They're just hanging out in the colon. They look kind of lonely. They're tired. But if you ate some broccoli, which does have some carbohydrate you would absorb, it would also come with some complex fibers, which you can't break down, you wouldn't absorb. So it would go to the colon, and look at these happy bacteria latching on to the fiber that got to feed them. And that would end up generating metabolites from breaking down the fiber. And one of the major types is called short chain fatty acids. That would get absorbed into the circulation. And I don't have time to go into all that, but that is the thing that we're looking for. Now, do all fibers or do all carbohydrates to get to the colon get used by the microbiome? No, not really. It's too early in this field to try to explain to you which ones would be best. The Sonnenburg families call these microbiota accessible carbohydrates, which the short term for is MACs, which kind of cracks me up because they are pushing Big Mac diets. I know that's a terrible McDonald's joke, okay, but not Big Mac like the hamburger, but high in microbiota accessible carbohydrates. So these are really two very different metabolic scenarios, one where you're getting a high fiber carbohydrate diet and one where you're just getting a lot of sugar and starch. One will feed the microbiome and one will not. So the goal is to get a lot of whole foods. That'd be beans and legumes, vegetables, fruits. So all the plant-based foods will come with fiber and feed your microbiome. Okay, now let's show you some, not cartoons, but some pictures. This is an electron microscopy picture that Justin actually won an award for by staining this cross section of the colon of a mouse. And so this very colorful set up here is staining all the different types of bacteria with different colors. This green thing running through the middle is the mucus lining in your colon that protects things from being absorbed into your cells. And all this blue coloring here, these are the nuclei in the cells in your colon. And what you really don't want is you don't want the nasty stuff in your gut that you've been eating to cross this barrier and get into your body. If they do, it could be something we call leaky gut syndrome. So back in the days when Justin was working with mice and these trillions of microbial cells, trying to figure out what they were doing, he did a mouse study where he gave some mouse a whole food plant-based diet, and he gave them a lot of sugar and jelly beans. I don't know how you give jelly beans to mice, but he found a way. And then he checked out what was happening in their colon. And so if this is the fiber-rich diet and this is a fiber-deficient diet, sorry, he's flipped these. So now the cells in blue are on the left and the bacteria on the right, unlike our first slide. This green thing here, this is the slimy green mucus. And I hope the point that you get here is that in the fiber-rich diet, this remained thick and intact, where this was thinned, making it more likely that the bacteria would sort of break through this barrier and get into our bodies. And one of the reasons it thins is this mucus lining, the components of it are carbohydrate rich. And so if you're not feeding the bacteria with carbohydrate from your food, they can eat the mucus lining in your colon and thin that. So that's what we would want to prevent. And here's a cartoon of it. Here's you getting a diverse high MAC diet with lots of foods. And here's the bacteria thriving and making short chain fatty acids, which can cross this mucus layer and get into our cells, which are pictured at the bottom. But you're taking supplements. The fiber supplements would feed some of them, but less of them. So you might get some thinning. If you weren't getting food and you're only taking one type of fiber, like inulin, a single fiber that you can get, that wouldn't support very many of the bacteria. This would thin even more. And if you're really not getting anything, then you've got a small number of bacteria making a small amount of short chain fatty acid. They're probably breaking through, leading to this leaky gut and inflammation. So that's the very small picture. Now let's go huge. Let's blow this up to the planet and across history and talk about some of the hunter-gatherer populations that Justin and Erica work with. So I think these are the Hadza from Tanzania. And he's thinking that, you know, for many thousands of years, this is what humans were doing. And then it was more agrarian. And now, very recently, it's become very westernized. So we've gone from lots of fiber to less fiber to no fiber in these fries where they took all of it out. What they find when they characterize the bacteria in the colons of these different populations. So if you go to all these sort of vestiges of hunter-gatherer populations that still exist, no matter where in the world they are, I don't actually know how to explain this graphic very well. It would help if Justin was here, but I know the main point was, no matter where in the world you are, the bacteria tend to be of the same type. And if you go to any Western country, whatever bacteria populated these groups is missing, and a different set of bacteria are populating those in the Western world. So here's another same graphic. This is the same thing from the last page, same set of countries, but now he's got a couple very specific families of bacteria. These three he finds all the time in traditional populations, not this one, and just the opposite for this particular family here as one example. If you really want a wonderful picture of what this looks like, in the traditional populations, Justin considers this a lush, tropical forest in the gut of the traditional populations and a barren landscape in a Western population eating a fiber-deficient diet. Or another way he frames this is in terms of thousands of years of those populations getting 100 to 150 grams of fiber a day, moving to an agricultural time in our lives when we had that shift, that agrarian shift, getting more like 35 grams a day. And then present day, really all the packaged processed food, taking all the fiber out before they ever feed it to us and getting a very low amount. So what I see as a nutrition scientist is this fear of carbohydrates. Oh my God, I'm on a low carb diet. Carbs do all these horrible things to you. Oh my God, I'm keto and paleo. I need more meat. I got to be more muscle bound. Low carb, high meat means low fiber. And so what we're seeing as a country is that we have dietary guidelines promoting 30 to 40 grams of fiber a day. We're getting from a group like the National Health and Nutrition Examination Survey reports that Americans only get 10 to 15 grams a day, when in fact we evolved with more like 100 to 150 grams a day. So we actually really have the capacity to do this. We've set kind of a low bar as a dietary guideline, and we're not even coming close to the low bar that we've set for ourselves. So he's done some elegant, he and Erica did elegant studies in mice, and I hope this isn't too complicated. I've tried to simplify this for you. One of the ideas was to take mice, and he gets these very fancy germ-free mice, so he has total control over the bugs in their gut. And so he gives them a high fiber diet to start with, and they start to develop a set of bacteria in their colon. And he takes one group and he keeps feeding them high fiber. He takes another group, he moves them to low fiber, and then after a while he switches them back to high fiber. And he does this across 4 generations of mice, one after the other, something you'd never be able to do in humans because this would take decades and decades and decades. So look at this mouse study where the idea is this is an abundance of different bacteria. And so each one of these little dots here represents some presence of bacteria. And I'll try to build this slowly. He's going to have 4 generations of mice, and one gets very little fiber and one gets a lot of fiber. And for every one of these generations, for those getting a low fiber diet, after being on low for a while, then he goes back and gives them high fiber. So the idea is if you compare the initial to this bar here, they went from high fiber to low fiber. They lost some diversity. He then re-fed them more fiber. And the point here is they gained some of the populations back, but not all of them. Now go to the next generation. Oh, they lost more, but you add some fiber and you get some back, but not a lot of them. And here's the 3rd generation, and here's a 4th generation. And the idea here is over multiple generations of this, which you can see quickly in a mouse population, they are losing a tremendous amount of diversity in their microbiome. And this is compared to a whole group of mice that were simply getting high fiber all the time, 4 generations of mice. So the concern here is that this elimination of dietary fiber not only depletes the microbiome that we have in our colon, but it's irreversible. You can't get it back by eating more fiber later. And so he's primed to work quickly before we deplete our microbiome even more than we have already. So we did a study where we were using pre and probiotics, and I'll share that with you before I conclude and take some of your questions. So our goal was to give dietary fiber prebiotics, fermented food probiotics, to live human beings instead of mice. We didn't chop off their heads and look in their colons. We only got to look at things in their blood. So there's things you can't do with humans that you can do with mice. As we started this study, we wondered, well, wow, there's a lot of different types of fiber, and there's a lot of different fermented foods. Wow, if we want to have 2 arms here, a pre and a probiotic arm, what type should we focus on and how much? And I hope you'll enjoy this, but this is how we often struggle as scientists. Okay, it might sound simple, pre versus probiotic, but there's a lot of choices here. How will we design this so it'll be relevant to the general public? And instead of just picking one, we said, heck, just get them to eat fiber, offer them grains and fruits and beans. And they could, as long as they can increase their fiber, we're happy. And we're not going to test one type of yogurt or one type of kimchi, kind of suspecting that different people might be averse to some fermented foods and like others. So let's just try to get them to eat as much fermented food as they can. We actually didn't have a set number. We thought if we had a set number, some people might be willing to go beyond that, but stop, because we had said, no, just this much. The thing that we had working for us was Dahlia, who we call the diet whisperer. Dahlia can get just about anybody to eat just about anything for a long amount of time. She's an incredibly inspirational health educator. So we got 36 people, 18 in each group, high fiber, high fermented, to take some baseline samples. And then we gave them 4 weeks to try to learn how to eat more and more and more of those foods. And then we asked them to keep that level for 6 weeks. And then we said, the study's over. Go ahead and eat whatever you want now, but we're going to check you at the end of this. And we did a lot of profiling for the immune system. So what we're really looking for is change the diet, characterize the microbiome, and then what do we see happening in the immune system in terms of protecting us from excessive inflammation. And this part is going to be very technical. We're going to have to go fast right now. Let me just ask you for kicks. How often have you gone to your physician and said, excellent, can I know my inflammation marker number? Can you just test that? I'd like my cholesterol, my glucose, my blood pressure, and my inflammatory number. Anybody been told their inflammatory number lately? So it's a very hot topic, immune function and inflammation, but it's actually very technical right now. We don't have a simple clinical value for you. But this is what we got them to eat over time, high fiber, high fermented food. So at baseline, our participants were eating about 20 grams a day of fiber and almost no fermented food. And then when we tried to get them to ramp up to a high level, we got them to double their fiber, and we got them to go from almost nothing to 5 servings a day of fermented food. Were they able to maintain that? Yes. For 6 weeks, they were able to maintain it. We're very pleased with our fabulous participants and thankful to Dahlia being our diet whisperer. And then this part's just amusing, a little bit anecdotal. When the study was over, we're actually quite pleased. There were no requirements after that. We wondered, will they be tired of the fiber and go back to the beginning? They didn't. They didn't maintain this very high level that we were promoting, but they sure are eating a lot more fiber on their own than beforehand. And similarly for the folks who weren't eating any fermented food, look at this. They're eating 2-3 servings a day now that the study is over. So here's what happened to the folks eating the fermented food is we saw a rise in diversity. These stars indicate the times when it was statistically significant during the maintenance period here, not statistically significant here, but this is the part where we were testing. The measure for this is quite complicated, but let's just call it alpha diversity. Over time, there are some diverse measurements. This is the one that's accepted in the field that we used. These are the number of observed types of bacteria going up. And of all the foods that we were feeding, the simplest ones that people liked were yogurt and kombucha. These were actually the most significantly related to the rise in the bacteria. I think that might be partly just because those were the most acceptable for people. So I'm not hanging my hat on that one yet. Over here were the types of bacteria in particular. This is a probability value of being important, and these are the 4 most important ones that increased. These changed as well, but these were the 4 most important families that changed. Now, you might expect if somebody's going to eat fermented food with bacteria in it, that your diversity would change because you would get the bacteria from the food you ate. So they actually went and bought all the different foods that people were buying in our study for the high fermented food. And they checked them out on these lovely Petri dishes. And they said, okay, what were the bacterial strains that people were eating in these foods? And then they did an analysis of the increase in the diversity of the microbiome to look at how many of them were in the food and how many were not in the food, but newly appeared in their guts. 95% of the new bacterial strains, the increase in diversity, were not found in the foods that they ate. This is actually pretty shocking. And I don't have a full explanation for this yet. And what the Sonnenburgs have hypothesized is that we actually harbor a number of different bacteria that are in such small amounts that they aren't typically detected. But if we change the environment of the gut, it might allow some of those to flourish and bloom where they weren't doing so before, and they were undetectable before. So it isn't simply the bacteria that you're eating from the probiotics. By just changing the milieu that's in our gut, changing that environment, it actually allows other bacteria to bloom and increase our diversity even more. So then we were looking from a clinical perspective of what would happen to immune function. So the name here is going to be immune profiling. And for an inflammatory state or a healthy state, some of these things should be high and some low. These are some technologies that they use in their laboratory. So they've color-coded them for us here. And for inflammatory markers in particular, for the fermented group, after looking at about 90 of these, about 18 of them, let's see, I better count them again, 5, 10, 15, 20, was that 22? Okay, so a lot of them, of the 90, were statistically significantly lower on the fermented food in purple. In fiber, there was only one that changed. It actually went up a little bit. So this was fascinating. Justin actually had expected the fiber group to do better. In fact, when we handed him the data, he wrote me an email and said, Christopher, did you happen to mix up group A and B? Because from all my mouse studies, I thought the fiber was going to do this. And from what you told me, it's the fermented food, but it must be wrong because we've been doing these mouse studies. In the mouse studies, it works the other way. I said, no, double checked. Group A is fermented and group B is fiber because he had been double blind while he was doing these experiments. So it was really the fermented food that led to a decrease in these inflammatory markers in the blood and not the fiber. Then he looked a little more closely in the high fiber group, and he had quite a few different ways to measure immune profiling, as I suggested. So he color-coded them for us here. 90 cytokines, 36 of these, 120 of these, 17 of these. And each one of these rows across is one single participant. And he used a statistical method that allows you to group them into groups that are more similar and less similar. And what he came up with, what pattern-wise from the colors looks like 3 different groups. This group and this group and this group, they kind of cluster together. Now picture this is about 9 people and 5 people and 6 people. These aren't huge groups. So we might be stretching it a little here. But this is a group whose inflammation actually got worse. And this is a group whose inflammation got better. And this group in the middle was in the middle. And what I found was that this particular group that had worse inflammation from the fiber had the lowest bacterial diversity to begin with. And we actually were checking in the stool to see how much of the fiber was actually being metabolized and used versus just being eliminated in the feces. And in these people, he saw quite a bit of the fiber was just passing through. It wasn't being used at all. And so the idea here, and this is their hypothesis, that this particular group actually experienced a little bit of harm from the high fiber. It's like they didn't have the bacteria to break it down, and giving them that huge challenge of fiber was a little too much for them. And it actually had a pro-inflammatory effect. Whereas this group at the bottom had the highest baseline diversity of bacteria, and they handled the extra fiber much better and lowered their inflammation. So in summary for our study, which we got published very recently, we got this high fiber group to eat very well for high fiber, shifted from some animal to plant protein, improved overall nutrient profile, changed their stool consistency, as you might imagine, no change in microbial diversity. They kind of had an individualized microbiota response. They increased in one type of bacteria in particular. They decreased something that I didn't have time to go into, but they didn't increase their fiber degrading capacity. Some of the fiber just passed through unfermented, and it looked like we had very much of an individualized response to the high fiber. And that was in contrast to the high fermented group. Everybody was eating fermented food. Some ate one, some ate another, but we saw pretty much increased microbial diversity across all of them. not necessarily from fermented food. And we saw decreased markers of inflammation across all of them. So this looked like fermented food did well for everyone, whereas the fiber was more individualized and more specific. Okay. So what we're after here is what kind of diet and lifestyle changes will change your microbiome, will lower your risk of chronic disease. So at the end, our whole research program right now is trying to combine what our group can feed them, what Justin can characterize with his fabulous wife, Erica, and then hand these all over to Mark Davis and Holden Maker, who run the Human Immune Monitoring Center at Stanford. And we think this is a trifecta moving forward for trying to understand this. What can you eat that will change your microbiome appropriately, that will improve immune function? Now, obviously, what I told you was not simple. There isn't a simple message here. So we're pretty early in this work, but we're very excited to be doing some human studies with these fabulous partners. And we just got our study of fiber versus fermented food published in one of the top scientific journals in the field. This is an altmetric score of what kind of visibility your article got. We're in the top 5% of all articles ever published from doing this. And this is just one of our initial studies in this area. We will be moving forward. And for anybody who wants any of these details, Sally, I will be happy to share the slides. So if anybody wants to review the slides, because I've gone too quickly to read over every bit, but hopefully people are catching the main point here. I want to thank, I'm not quite done. I have a little bit of wrap up to go. I'm going to thank these fabulous grad students and postdocs from the Sonnenburg lab who wrote all this up. I want to thank all the fabulous team members that recruited humans, held their hand, collected all their samples. And actually the Sonnenburg lab and our Gardner nutrition lab is even bigger than that. Thanks to lots of them, but I want to leave you with some conclusions about feeding your friends, and that peril may lie ahead. Please don't get ahead of the science. I think for me, scientifically, this is all very sexy and fun and catching lots of headlines. Our study was actually in the New York Times 1 week, and they got such a strong response from it. They had a follow-up article in the New York Times the next week telling you how to get fiber and fermented food in your diet. So very popular public approach here. The vision is that maybe we can have some microbiota-based medicine. Maybe we can use this variability and malleability of the microbiota to improve human health. It may be that someday you go in and they type you for your microbiome and they figure out a way to target something that you need, and we get microbiota-based precision medicine. The challenge is that everyone has their own unique microbiome. There's another group in Israel led by Eran Segal. This is a beautiful paper where they got 800 people to eat a lot of different things. And then they did machine learning, and they have an app for you now that tells you how to predict your glycemic response to specific foods. This would be of great interest to people who have prediabetes or diabetes. There's a lot of movement here, but the science still isn't perfect. So be careful. 4 take-home messages would be: our Western gut and our Western diet is problematic. We don't have enough diversity. We're not getting enough fiber or fermented food. Dietary fiber has some benefits, and now I've explained what prebiotics are. This isn't just a result from our study. This is looking across the whole field. Looks like dietary fiber is good. Looks like fermented foods are good. These are things that a lot of people don't eat. Why not? The challenge is you're going to find a lot of claims. Oh, take my fiber. Oh, take this probiotic pill, and this thing will happen to you, which we don't actually know yet. We need more studies. But please do remember to feed your friends so that you don't break through that mucus layer, which means prebiotics and probiotics. I prefer to think of food instead of pills. So if you wanted some fiber and fermented food, think about delicious Korean kimchi. So look at all the fiber in that Middle Eastern vegetable salad, Mediterranean roasted veggies. I make kombucha at home. I'm on like my 70th batch. I make a batch of kombucha every week. It's pretty easy, and I really like it. My friends like it. A hummus sauerkraut salad yogurt, nuts, and berries, fiber and fermented food. Enjoy. Thanks for the opportunity to share some of this with you.
This was amazing, Professor Gardner, and I would love to get a copy of your slides. For those of you who were asking, we will share the recording, but because this is dense, yes, if we can get a PDF of the slides, I'll include that with the recording.
I will.
This was just on so many levels, and people are typing into the chat. This was so good. I'm going to go through some of these questions. One quick one that I had personally, so I'm going to sneak it in, is we look at probiotic foods like yogurt, kombucha, kefir. Is there a benefit to eating a variety of probiotic foods versus just yogurt, or don't we know that yet?
So we're assuming there's a benefit for the variety because it's sort of like I actually work with another group called Zoe that has 1,000 twins in the UK, and they have studied their health and their microbiome to see how much of our health is determined by genetics and how much environment and how much can you change your microbiome because you can't change your genetics. They've actually done a wonderful job of trying to specify specific bacteria that are positive and specific bacteria that are negative. It's one of the first groups that I've seen that's come up with a list of the specific bacteria. There isn't consensus on that list. Those lists are just starting to emerge of what the best and the worst will be. But Sally, the challenge here is going to be there are trillions of bacteria in your gut that come from hundreds of species. And to think of all the permutations there are, just because you might be deficient in one that has consistently been found to be healthful or not healthy, doesn't mean just focusing on that one would help. It seems silly to think that in the milieu of everything else that's in your gut, that one would be the answer. So until we do more, I think the simplest answer is diversity of fiber and diversity of fermented food. Obviously, something that you need to like. Let me just give you a really quick example is somebody took this and yes, I totally want to do this, but I hate kimchi. I said, how many times have you tried kimchi? Once, it was too spicy. Oh my God, there are hundreds of kinds of kimchi. Some are spicier and some are less spicy. So, try some of these different things. Somebody says, yes, yogurt. Oh, I can eat yogurt. I'm going to go get my peach, guava, pineapple, low-fat yogurt from the grocery. I said, oh, did you see how much sugar is in that thing? That's not yogurt. That's like a sugar delivery system. Get some plain yogurt and put your own berries and granola on it, which will provide fiber with your probiotics.
Yeah.
So diversity for now.
Yeah. I love the advice about, find something you like. And I know there were questions that came in through the chat about how to make your own. I'll include this link in the follow-up material, but Stanford Healthy Living Program is having a special program with a chef on how to make your own fermented foods. So I'll include that because I think it's a good find. All right, let me get through some of these questions. Arthur, bless your heart, I loved yours. I'm going to start with yours. So remember, Professor Gardner, how we were showing people that, started with the low carbohydrate, low fiber diet. They were kind of hosed in your study results. So Arthur says, are you saying that if I am 80 years old and have been on a low fiber diet for my whole life, I might as well give up? What would you advise here?
I can't believe you're alive. You're on a low fiber diet and you're still alive. Well, you're obviously doing something right. So Arthur, this gets back to how diverse humans are. So you're obviously doing something right if you're 80 and you're listening to this because you're interested in your health. So none of this is the end all be all. It's just an opportunity. In all of our studies, Arthur, we have outliers on both ends, and most of our messages are targeted to the general public for an average response, but everybody does respond differently. And there's going to be nothing more true than this microbiome thing because everybody's microbiome is so different. So I don't have a good answer of why you're doing well. I can say that if you listen to this and you rushed out and you bought a lot of fiber and it caused you discomfort and gas and and you didn't enjoy it, then there's no reason to do it. You are doing fine.
Yeah.
to begin with.
Wait,
But for those who have issues, for those who have health issues, we think this approach will help a bunch of people that are in that category, although we're not quite sure exactly what to tell them to do.
yeah, because it's a new field. And so my research at USC is looking at what happens in older populations. And so we see that older adults have less diversity than their younger counterparts. So even trying some yogurt every day might be good based on what you found, Professor Gardner, about how probiotic foods can contribute to diversity, even in a short time. I think your study was 17 weeks. Am I right?
Yeah, only 4 weeks to ramp up and 6 weeks on it. So another thing, yeah, you're exactly right. What if people did this for a year or 2 years or 3 years? We don't have those studies because it's really hard to get somebody to sign up for a human study and say, hey, just for kicks, can you change? Christopher's curious. Will you do this for the next 5 years or 5 decades? No, I'm willing to do that for 6 weeks. So we'd have to do longer studies.
Yeah, yeah. Here's a question about the FODMAP diet. So some people, and one question in particular, people who have difficulties either with fermented foods or they have difficulties with high fiber foods. What advice might you have for somebody who is at least for, currently somewhat limited in terms of the fiber rich foods they can eat?
Yeah, well, look, and this is me. So I'm 62 years old now, and I keep changing my diet, but slowly over time. I have a really solid foundational diet, and I learned something new. And instead of getting so excited that I overdo it to the point where I just can't take it anymore, do one of those SMART goals that's measurable. I'm going to try eating yogurt twice a week. I'm going to try 3 kinds of kimchi this month and see if I can build it into my regular diet. One of my personal changes in life because of studying with Justin is I now have avocado toast with kimchi on it twice a week. That is one of my staple breakfasts. And I had avocado toast for many years, not with kimchi on it. And it is delicious. I know it's not for everybody, and I don't have it every day. I only have it twice a week. And that's something that's super manageable for me. And I enjoy it. So trying to find some of those, and if they fail, don't continue, try something else.
Yeah. I've been motivated by my research and what I've read with your research. And something recently I've tried that you guys might enjoy is I take a champagne glass, I put it in the freezer, and at night I have a champagne glass with kombucha because it's bubbly, it's sparkly, it's fun. Yeah. And, I really don't drink much alcohol, but I have kind of a good time with it.
Woohoo.
All right. So here's a question. How do you feel about luncheon meats that have no preservative, nitrates, or colorings? Are they still considered dreaded processed foods? So I realize that's a little bit off topic, but it was an interesting question. Should we avoid lunch meats?
Yeah, again, it's a continuum. What are you having it with? Are you having it, I need white bread to put between this, and I need something really I need butter on it or something like that. And I was going to have a lentil salad, but instead I had this sandwich meat between 2 pieces of white bread. That'd be really different between I've got this fabulous whole grain bread and I've got some leafy greens that I like on this. I've got some stone ground Dijon mustard on this thing. And I really enjoy it more if I put a slice of regenerative farming, ranching, no nitrate, everything free. And otherwise, I was going to go to McDonald's and have a hamburger, but I like this. So Sally, in my world, I'm always looking for instead of what and with what. So I can see making that lunch meat healthier or unhealthier, depending on all the other contexts that go with it. So all these foods are on a continuum. So if you had, a fresh cut piece of meat versus a very highly processed hamburger, we can find ends of the spectrum there. And it sounds like your listener is somewhere in the middle. It's not the healthiest and it's not the least healthy, and made some movement in the right direction.
And it's about making better choices most of the time. Not all the time, overall. Yeah. I'm going to take 2 more questions. We might run just a couple minutes over. Are you okay staying on the phone? Okay. So I think this was good. You kind of addressed it, but Mary wanted to know about Probiotic supplements, are taking those supplements worthwhile? And what about fiber powder? So, what do you feel is the benefit of eating it in a real food versus taking it as a supplement?
And let me start with the probiotic supplement. And one of the ideas there is I don't think we have a lot of great data on how many bacteria are surviving, right? So maybe when they put it in the bottle, it had 40 billion CFUs, but how long has it been there? And has it really got the species it says it does? That unfortunately, the supplement industry is not very well regulated. And if you go back to vitamins and minerals, there are many examples where they don't actually have what they say they had on the label. And there's many where they do. There's many above board, great companies. But I'm a little worried when you do that. We've actually done a study recently where somebody suggested 3 different bacteria in a probiotic supplement. And we said, why those 3? There's hundreds. And they said, well, we think these are the 3 best. Well, I don't know. There's so many possible permutations there that we don't know if those are the 3 best. And if it's only one, we really don't know. As for fiber supplements, that can be an easier way to digest it. Maybe you can't stomach all the different foods and the bulk of the lentils and the beans and the vegetables. So it's a way to add some fiber. But most of the fibers are very simple. Inulin from chicory root is one of the most popular right now, or Metamucil from psyllium. But that's really just a single type of fiber. And so the chances that it's going to be beneficial are limited because of that, which is why for now, we're promoting variety. Is that better than nothing? Yes, it's better than nothing.
Yeah.
But hoping to push people toward diverse food choices.
Yeah, because there's also so many other health benefits of eating the rainbow, not just the gut health. So let me end with this last question, but then for everyone, you're going to get the slides, you're going to get the video, you're going to get some key links. How do I know? How would I know if I have a healthy gut? It's like, because we're not taking pictures like the Sonnenbergs did of those mice.
So this is, okay, and this is the one, a great way to end on: beware the perils of going beyond the science. So there's multiple companies out there that if you pay them, they will characterize your microbiome and send it back. One of those companies is being sued in court right now for false representation and for misleading claims. So I think it costs about $100. You got to put your poop on a stick and stick it in a tube and mail it back to them and pay them. I'll tell you that the deep secret here is one of the things those companies are doing is taking your money and finding things out. They're asking you other questions and they don't actually know the answers yet. But if they get more of your poop and ask you questions and find out what's going on to you, that's one of the ways we'll learn what this is all about. The company that I work with, and I don't get any money for this, so this is not trying to promote it for any personal gain here, but Zoe is a company in the UK, and they actually have some advice for you based on your microbiome, but they are very actively collecting data on people and using this as a way to build a platform to understand more about, as one type of bacterial strain goes up or down in your gut versus another, what are they seeing in terms of glucose or cholesterol or health or outcomes? So to flip it around, although don't count so much on what people are saying right now, if you do participate in this, we will know more. We actually need thousands of people to provide data so that we can see what subsequently happens to folks. I just wouldn't put a huge amount of weight on the advice that we have today, but unless we do this, we won't know tomorrow any more than we know today.
Yep. And someone just asked, how can we be apprised of future studies? If you're looking to recruit people, I think some of our participants might be interested. Is there a link you can share, or do you want to just reach out to me if you're doing a recruiting?
So I will say that this company Zoe is doing something right now, ZOE. You can look for microbiome and
Cool.
Zoe. They're recruiting people into a study. You actually, I can't remember what the deal is about paying for an app, but you get results back from the Zoe study. They don't know exactly what to tell you, but they're looking how to find out. Sally, in my lab, Justin and I are doing much smaller things. They actually did a study with 1,000 twins, and then we helped them do a study trying to see if we could recruit 1,000 people remotely without bringing them in, and it worked really well. So they're now trying to recruit 10,000 people. and have them do this over time. And that study has been initiated. It's called Predict 3. The first one in twins was Predict 1. The remote test was Predict 2. And they've now moved on to Predict 3, where, as the name implies, they're trying to predict what this will do to help you.
Perfect. Someone just typed it in the chat, and I'll include that link as well. We've kept you over time. Professor Gardner, this was so interesting. Thanks to you. Thanks to everybody who joined us. And happy Wellness Wednesday. Bye now.
Excellent. Okay, I'll try to Twitter some of this stuff when I find out what it is, at Gardner, PhD.
Okay, thank you so much.
All right, Sally, see you next time.
Bye
Nice
now.
to meet you all remotely.